I'm sure we've all committed to something that we've later come to regret. It could have been a house, a car, or maybe even a tattoo. But unfortunately, regrets also happen in manufacturing one of the most lucrative classes of drugs in history: monoclonal antibodies like Humira, Keytruda, and Stelara. Behind each of these drugs is a long biomanufacturing process that begins with something called cell line development. This is the process of engineering a cell to make a biologic of interest. Cell line development is important for two main reasons. First, the FDA requires that every batch of every dose ever administered over the lifetime of a drug come from a single cell's descendants. Second, and because of this, the productivity of this cell line can dictate up to 50% of the cost of drug manufacturing. Throughout customer interviews as part of the I-Corps program, we repeatedly heard how important productivity was in biomanufacturing, with one customer even saying, "In biomanufacturing, titer is king." But there's a problem. Clonal cell line productivity is variable and unpredictable. To find a highly productive cell, companies will often screen at the earliest stages of the biomanufacturing process. But even if a cell looked productive and promising in its infancy, it can lose its performance over time, leading to regret. Biolinco is developing a high-throughput platform that can link a cell's past to its future using a proprietary DNA barcoding technology. We are the only company able to track productivity over time, enabling companies to base decisions on real manufacturing data instead of guess using early-stage performance. Altogether, we can save companies up to $120 million annually per drug by barcoding clones, tracking the barcode productivity, and then going back in time to select the most productive clone. To best serve our customers, we intend to operate as a traditional contract development and manufacturing organization, or CDMO for short. In this business model, customers will provide us with the drug that they would like to manufacture, alongside an upfront payment to cover the initial cost of cell line development. We'll collect additional milestone payments as we achieve pre-specified goals. Biolinco will then apply its high-throughput platform to screen thousands to millions of cells for the most productive cell line, ultimately isolating a master cell bank with the most productive clone. This bank will then be returned to the customer for large-scale manufacturing and subsequent patient treatment. We will also collect royalties on a small percentage of the profits generated from the use of the cell line. We envision a stepwise approach to growth over the coming years. During product development, we will have to work with a model monoclonal antibody in order to demonstrate the utility of our platform. As a result, after we achieve our minimum viable product, we will have successfully developed a biosimilar cell line, which can then be licensed to companies for immediate cash flow. After our platform becomes trusted, we can expand into other CDMO services, including cell line development for novel monoclonal antibodies, bispecific antibodies, as well as critical quality attribute testing and tuning. Finally, we envision becoming a fully GMP-certified CDMO with end-to-end biomanufacturing services, as well as services for up-and-coming therapeutic modalities, such as adeno-associated virus and lentivirus. We know there's a strong demand in this sector based on our relationships with AMBIC, or the Advanced Mammalian Biomanufacturing Innovation Center. This is a consortium with more than 30 biomanufacturing companies that regularly come together and support research that addresses pain points collectively faced by the sector. To this end, we have successfully raised $200,000 to support underlying technology development from AMBIC, as well as obtained a letter of support from Sigma, indicating their willingness to test our technology with further development. To protect our platform, we've submitted a provisional patent that contains both composition of matter and method of use components for essential process components. In the next 12 months, we expect to file for a full patent and obtain an exclusive use license from Johns Hopkins Technology Ventures. Of course, we're not the only company operating in this space. However, we do expect to meet or exceed industry standards across a variety of metrics, including titer, cost, turnaround time, and labor. However, the most differentiating factor is likely the number of stable clones we can screen at a late stage of the biomanufacturing process, surpassing our competitors by several orders of magnitude. Additionally, because cell line development takes place largely before FDA involvement, there are no direct regulatory filings required, speeding up development and reducing time to market. After we obtain GMP certification, we will be able to provide companies with full assistance with their IND and BLA filings as necessary. To date, we have received more than $300,000 to support proof of concept work for Biolinco's platform. As we look to the future, we are working to raise additional non-dilutive or pre-seed and seed funding to support prototype development over the next year. In 2026, we expect to obtain our first sponsored research contract or pilot research contract with customers, with a goal of full commercialization in 2027 and beyond. Our team consists of scientific experts with close ties to the biomanufacturing sector. I'm Kent, and I defended my PhD 2 weeks ago in Michael Beatonbaugh's lab. I'm still very excited about that. Thank you. Mike Beatonbaugh is the director for the Advanced Mammalian Biomanufacturing Innovation Center, and I will be continuing my work with Biolinco as a postdoc in Reza Kalhor's lab. My other co-founder, Jinan Wen, is in the last year of his PhD in Mike Beatonbaugh's lab and will join Biolinco full-time after defending. Altogether, we are developing a high-throughput cell line development platform, leveraging innovative DNA barcoding technologies, and we want you to be a part of the cell line development revolution. So join us as we eliminate regrets from the biomanufacturing process.